don't stop. Don't stop. Don't stop. Don't stop.
Chapter 164. Autonomic Nervous System, ANS. Introduction. Autonomic Nervous System, ANS, is primarily concerned with regulation of visceral or vegetative functions of the body. So, it is also called vegetative or involuntary nervous system. Divisions of ANS. From anatomical and physiological point of view, ANS is divided into two divisions: sympathetic division, parasympathetic division. Differences and comparison between both the divisions of ANS are given in tables 164.1 and 164.2. Sympathetic division. Sympathetic division is otherwise called thoracolumbar outflow because the preganglionic neurons are situated in lateral gray horns of 12 thoracic and first two lumbar segments of spinal cord. Fibers arising from here are known as preganglionic fibers. Preganglionic fibers leave the spinal cord through anterior nerve root and white rami communicantes and terminate in the postganglionic neurons, which are situated in the sympathetic ganglia. Sympathetic division supplies smooth muscle fibers of all the visceral organs such as blood vessels, heart, lungs, glands, gastrointestinal organs, etc. Sympathetic ganglia. Ganglia of sympathetic division are classified into three groups: paravertebral or sympathetic chain ganglia, prevertebral or collateral ganglia, terminal or peripheral ganglia. Paravertebral or sympathetic chain ganglia. Paravertebral or sympathetic chain ganglia are arranged in a segmental fashion along the anterolateral surface of vertebral column. Ganglia on either side of the spinal cord are connected with each other by longitudinal fibers, to form the sympathetic chains, Fig 164.1. Both the chains extend from skull to coccyx. Ganglia of the sympathetic chain, trunk, on each side are divided into four groups: cervical ganglia, 8 in number; thoracic ganglia, 12 in number; lumbar ganglia, 5 in number; sacral ganglia, 5 in number; cervical ganglia. 8 cervical ganglia are arranged in three groups: superior cervical ganglion, it is formed by the fusion of upper 4 cervical ganglia. It is the largest ganglion of ANS. It receives preganglionic fibers from first thoracic spinal segment, T1, via white rami. Postganglionic fibers from this ganglion supply the blood vessels, glands, etc. Superior cervical ganglion also sends some fibers to heart through superior cervical sympathetic nerve and cardiac plexus. Middle cervical ganglion, it is formed by 5th and 6th cervical ganglia. Preganglionic fibers arise from T1 segment. Postganglionic fibers from here supply the sweat glands, thyroid gland, and parathyroid glands. It also sends fibers to heart via middle cervical sympathetic nerve and cardiac plexus. Inferior cervical ganglion, this ganglion is formed by the fusion of 7th and 8th cervical ganglia. First thoracic ganglion fuses with inferior cervical ganglion, forming stellate ganglion. It receives preganglionic fibers from T1 segment. It sends postganglionic fibers to heart through inferior cervical sympathetic nerve and cardiac plexus. Postganglionic fibers also form the plexus around subclavian artery and its branches. Thoracic ganglia. There are 12 thoracic ganglia on each side, and these ganglia are evenly spaced. Thoracic ganglia receive preganglionic fibers from the thoracic segments of spinal cord. Postganglionic fibers from thoracic ganglia are distributed to visceral organs in the thorax and abdomen. Lumbar ganglia. There are 5 lumbar ganglia. Preganglionic fibers for these ganglia arise from first and second lumbar spinal segments, L1 and L2, and reach the lumbar ganglia. From here, the fibers extend down to sacral ganglia also. Postganglionic fibers from these ganglia supply the abdominal and pelvic organs. Sacral ganglia. There are 5 sacral ganglia, which receive the preganglionic fibers from L1 and L2 segments. Postganglionic fibers from sacral ganglia innervate the blood vessels and sweat glands in the lower limb. Below the sacral level, both the sympathetic trunks converge and fuse upon the anterior surface of coccyx and form a terminal swelling. This terminal swelling is known as coccygeal ganglion. Unpaired coccygeal ganglion is also called ganglion impar. It receives preganglionic fibers from L1 and L2 segments. Postganglionic fibers from here are distributed to the abdominal viscera and pelvic region. Prevertebral or collateral ganglia. Prevertebral ganglia are situated in thorax, abdomen, and pelvis, in relation to aorta and its branches. Prevertebral ganglia are celiac ganglion, superior mesenteric ganglion, inferior mesenteric ganglion. Prevertebral ganglia receive preganglionic fibers from T5 to L2 segments. Postganglionic fibers from these ganglia supply the visceral organs of thorax, abdomen, and pelvis. Terminal or peripheral ganglia. Terminal ganglia are situated within or close to structures innervated by them. Heart, bronchi, pancreas and urinary bladder are innervated by the terminal ganglia. Sympatholoadrenergic system. Sympatholoadrenergic system is a functional and phylogenetic unit that includes sympathetic division and adrenal medulla. Adrenal medulla is a modified sympathetic ganglion. Since adrenal medulla and sympathetic division develop from the same neural crest, their secretions and functions are almost the same. Any increase in sympathetic activity increases the secretion of catecholamines from adrenal medulla, refer chapter 71. Parasympathetic division. Parasympathetic division of ANS is otherwise called the craniosacral outflow because the fibers of this division arise from brain and sacral segments of spinal cord. Cranial outflow or cranial portion of parasympathetic division. Cranial outflow or cranial portion of parasympathetic division arises from brain stem. It innervates the blood vessels of head and neck and many thoraco-abdominal visceral organs. Cranial outflow includes the following cranial nerves: oculomotor, 3, nerve. Facial, 7, nerve. Glossopharyngeal, 9, nerve. Vagus, X, nerve. Preganglionic fibers of these cranial nerves arise from neurons situated at two different levels: tectal or midbrain outflow, 3 cranial nerve. Bulbar level or bulbar outflow, 7, 9, and X cranial nerves. Preganglionic fibers are longer and reach the postganglionic neurons, which are situated within the organs or close to the organs innervated by these nerves. Preganglionic fibers are myelinated, but the postganglionic fibers are non-myelinated. Tectal or midbrain outflow. Group of cells forming Edinger-Westphal nucleus of 3 cranial nerve gives rise to tectal fibers. Fibers from this nucleus end in ciliary ganglion. Postganglionic fibers from here supply the sphincter pupillae and ciliary muscle. Bulbar level or bulbar outflow. Preganglionic fibers are the fibers of 7, 9, and X cranial nerves, which arise from the nuclei present in the medulla oblongata. Fibers of 7 cranial nerves supply the lacrimal, nasal, submaxillary, and sublingual glands. Preganglionic fibers of this nerve end in sphenopalatine ganglion and submaxillary ganglion. Postganglionic fibers from sphenopalatine ganglion supply lacrimal and nasal glands. Postganglionic fibers from submaxillary ganglion supply sublingual and submaxillary glands. Fibers of 9 cranial nerve supply the parotid gland. Preganglionic fibers synapse with neurons of otic ganglion. Postganglionic fibers from otic ganglion supply the parotid gland. Fibers of X cranial nerve supply visceral organs of the body. Preganglionic fibers terminate in the ganglia, which are situated on or near the organs. Postganglionic fibers from the ganglia supply the organs. Vagus nerve supplies almost all the organs in the thorax and abdomen, but not the pelvic organs. Sacral outflow or sacral portion of parasympathetic division. Sacral outflow or sacral portion of parasympathetic division arises from the sacral segments of spinal cord. It innervates smooth muscles forming the walls of viscera and the glands such as large intestine, liver, spleen, kidneys, bladder, genitalia, etc. Preganglionic fibers arise from anterior gray horn cells of second, third, and fourth, from first also in some cases, sacral segments of spinal cord and form the pelvic nerve, nervi erogens. Fibers end on postganglionic neurons, which are situated on or near the visceral organs. Fibers from postganglionic neurons supply descending colon, rectum, urinary bladder, internal sphincter, urethra, and accessory sex organs. Sacral parasympathetic fibers supply those visceral organs which are not supplied by vagus. Functions of ANS. Autonomic nervous system is concerned with the regulation of functions, which are beyond voluntary control. By controlling the various vegetative functions, ANS plays an important role in maintaining constant internal environment, homeostasis. Almost all the visceral organs are supplied by both sympathetic and parasympathetic divisions of ANS, and the two divisions produce antagonistic effects on each organ. When the fibers of one division supplying to an organ is sectioned or affected by lesion, the effects of fibers from other division on the organ become more prominent. Actions of the sympathetic and parasympathetic fibers on various structures are given in Table 164.1. Neurotransmitters of ANS. Different nerve fibers of ANS execute the functions by releasing some neurotransmitter substances, Table 164.2. Sympathetic fibers. Preganglionic fibers, acetylcholine, ACH. Postganglionic noradrenergic fibers, noradrenaline. Postganglionic cholinergic fibers, ACH. Postganglionic sympathetic cholinergic nerve fibers supply sweat glands and blood vessels in heart and in skeletal muscle. Parasympathetic fibers. Preganglionic fibers, ACH. Postganglionic fibers, ACH. Catecholamines. Synthesis and the metabolism of catecholamines are explained in Chapter 71. Acetylcholine. Refer Chapter 141 for details of acetylcholine. Receptors of neurotransmitter. Adrenergic receptors, details are given in Chapter 71. Acetylcholine receptors, details are given in Chapter 141. Sympathomimetic drugs. Sympathomimetic drugs or adrenaline-like drugs are the drugs, which produce the effects of sympathetic stimulation. Adrenaline and noradrenaline produced in the body act only for a short duration of about 1 to 2 minutes. Whereas, sympathomimetic drugs injected intravenously act for a longer period of about 30 minutes to 2 hours. Sympathomimetic drugs are drugs stimulating the receptors directly. Phenylephrine, alpha receptors. Isoproterenol, beta receptors. Albuterol, beta 2 receptors. Drugs inducing the release of noradrenaline. Ephedrine tyramine amphetamine. Sympathetic blockers. Sympathetic blockers are the drugs that prevent actions of sympathetic neurotransmitter. Sympathetic blockers act on all levels. Actions of blocking agents are given in Table 164.3. Parasympathomimetic drugs. Parasympathomimetic drugs or ACH-like drugs are drugs, which produce the effects of parasympathetic stimulation. ACH produced in the body acts only for a short period, whereas the injected ACH acts for a long time. Similarly, parasympathomimetic drugs also exhibit their actions for a longer time. Parasympathomimetic drugs are drugs which act on muscarinic receptors. Pilocarpine and methacholine produce their effects by acting on the muscarinic receptors. Drugs which prolong the action of ACH. Action of ACH can be prolonged by preventing its destruction. Drugs like neostigmine and physostigmine inhibit the activity of acetylcholinesterase, and so the ACH is not destroyed quickly. Parasympathetic blockers. Parasympathetic blockers are drugs which prevent the actions of parasympathetic neurotransmitter. The drugs atropine, hematropine, and scopolamine inhibit the actions of ACH by blocking the muscarinic receptors. Ganglionic blockers. Ganglionic blockers are the drugs that prevent the transmission of impulses from preganglionic neurons to postganglionic neurons. Tetraethylammonium ion, hexamethonium ion, and pentalinium are some of the ganglionic blockers. These drugs block both sympathetic and parasympathetic ganglia. However, ganglionic blockers are commonly used to block sympathetic ganglia, rather than the parasympathetic ganglia because sympathetic blockade overshadows the parasympathetic blockade.